had the ability to use fluorescent tags on the dideoxynucleotides so that 4 lanes, or in this case 8 lanes, could be coalesced into a single lane just going by color, which is what I showed you last time on that chromatogram handout with all the different colors. Before that, we had to use radioactivity. And radioactivity, every single lane was black. So you couldn't put them all in one lane. You had to have 4 different lanes because you needed to have a G reaction, an A reaction, a T reaction, and a C reaction to be able to distinguish them. So this particular gel happens to be loaded in the order G, A, T, C, A, C, G, T. It's double loaded. So you have the G reaction loaded twice, for example. You have the A reaction, the exact same reaction, loaded twice. So this lane is the same as that lane. This lane is the same as that lane. So if you look at G, A, T, C, A, C, G, T, it is in this order because G is next to an A, G is next to a C, G is next to a T, A is next to a G, A is next to a T, A is next to a C. So every nucleotide is next to every other nucleotide for comparison, because otherwise, if you only have the first 4, G, A, T, C, there might be a place where you're like, well, I can see that there's a G and I can see that there's a C, but maybe it's up here and I can't see which one's higher than which one because they're really kind of close. Well, then you go to the place where a G is right next to a C, and because they're right next to each other, you'll be able to see which one is higher. So this means that when you're reading this sequence, you're not doubling up what you're reading. You're reading GATC and then just realize it's loaded the same over here. So in this one, for example, there's a little line which goes all the way across. This is where you're going to start from. It's actually a G. You can sort of see a G right here. You'll see this on all 3 films. I have 3 films for 5 groups, so you might have to share a little bit or pass it off. So this lane, which kind of goes all the way across, that's a good starting point. You're going to want to read about 50 bases up to be able to put into the BLAST search. And so when you're doing this, after this initial lane right here, remember this is GATC. So this is a CC right here. There's 2 bands. So you read this as CC AAG. Okay, not CCCC GGGG. I'm sorry, AAAAGG, because this is doubled over here. So it's just CCAAG. You can sort of pretend that half doesn't exist until you need to confirm with a G being next to a C or something like that. So what I find helps is taking a little index card or a piece of paper and writing GATCACGT on it and moving it up the film that you'll have. Do not put a pen on my film. Do not make any marks on my film at all. I've had people making little marks in the past, and it annoys me. So because this film gets used every semester. So basically, you want to start from here and read about 50 bases up. If you read about 50 bases up, your mutation sequences will be contained in that area of about 50 bases. So this is your wild type sequence. And you're going to compare the wild type sequence with the mutation sequence that I handed you last time. And so that row up at the top. Now remember, I said you're not going to start right at the very beginning, because at the very beginning is a whole bunch of ends. So maybe start around 15 to 20 bases in whenever you think the sequence starts to become very, very clear. Probably not more than 20 bases in though, unless you have a real problem. Also remember that after 20 bases, if you have an N, you can resolve this by eye. You are smarter than the computer. You don't have to, say, does it fit into my predetermined algorithm for, which lane is higher? You can see that, okay, maybe an A and a T are really close, so the computer couldn't call it, but I can see that there's an A and a T here, and it lines up with, an A and a T on the gel. Remember, there are no additions, no deletions, only actual mutations here. So you might need to read this, put it in the computer. The computer is going to tell you in the BLAST search that I mentioned in the homework page. You'll go to that website, you'll put in the 2 sequences, and maybe you'll have, say more than 2 areas. But once you find your 2 areas of mutation, you're good. I'm not asking you for 3 areas of mutation. And also remember my parameters last time. If there's only 1 base between some mutations, that's all the same area. If there's 2 bases or more between mutations, that's a different area. So some of the mutations are closer to each other, some are farther apart. You have different possibilities for the different groups. And I will be here if you have any questions. We have 3, and then after you have done the alignment in the BLAST program, you're going to take the wild type and you're going to put it into BLAST, and when you do that, it'll bring up a whole bunch of possibilities of, okay, and you remember to check Gallus gallus when it asks you what organism. Gallus gallus domesticus, it's a domestic chicken, and it'll come up with maybe a whole bunch of possibilities for U4 and UX. And if you find one that says gene, you can click on the gene, and it should tell you what chromosome it's on, or in that area. I think you'll see it. You'll see something in that area when you start scrolling down possibilities. But I will be here for questions. So we have a light box here you can read. We have a light box over there you can read. You can put it on an iPad, which is white, and get a good and use that as a light box. There are different possibilities. Okay, so come and get one of the 3. I'll be here for questions.
And have fun with this, you guys, because understanding the foundations of how DNA used to be read is really important to understand.
Yeah, once you read a radioactive gel, you will really appreciate how it's done so much easier now.
Yes. Trust me, you'll appreciate technology.
Yeah.
So,